Hello, everyone. Today I get to speak with Dr. Anna Urciuolo, who has invented a way to do 3D intravital bioprinting. Wait till you hear. Dr. Urciuolo, thank you so much for taking the time to speak to me today.
Thank you to you. It's a pleasure.
Would you introduce yourself?
Yeah, I'm Anna Urciuolo, and I'm a scientist. I'm a mom. I'm a woman. Here I am.
And you recently had a paper published in Nature Biomedical Engineering entitled Intravital Three-Dimensional Bioprinting.
Yes, we hope that this will open new perspective to be applied then in vivo in patients.
So my naive understanding of the paper is
Yeah.
that you can 3D print biomaterials inside of a body without surgery, that is, without cutting open the skin.
Yeah, actually, this depends on the area in which you are working on.
Mm-hmm.
So basically, if you are able to pass through the tissues, as for example in the skin, what we did was to inject under the skin our hydrogel that was in a liquid form. And then by using the multiphoton light, we were able to create the structure according to the design that we defined before. So one important step is that we combine the intravital microscopy to the bioprinting system. Basically, by using this light, you can reach your sample inside the tissue because this can pass through it. It's basically the same approach that people are using for doing intravital imaging. Mm-hmm. But instead of imaging, we can bioprint. And this helps also us to have an idea exactly of where we are because we can couple the bioprinting with the imaging.
Interesting.
So we can imaging until we are bioprinting, and we can imaging before bioprinting, we can imaging after bioprinting.
What is the substance that's inside of the body? What is this hydrogel and how does it relate to stem cells? I know in the paper you discussed stem cells a bit.
Yeah, so basically the most important achievement was to find out this compound that can easily be clicked. So by using the infrared light. So differently from other hydrogel that are photosensitive, in this case, what happens is that you don't need any other additional chemicals to create the click chemistry. So the click and the bound among the different polymers that you have in solution. So in this way, we don't have any radical reactions or reaction that can harm somehow the body or the tissue itself. It's a very simple chemistry, basically. And what happens is that when the light is reaching the hydrogel, the molecules can be activated and a new bond can be formed. So all the polymers that are attached to the molecule can then join together in a structure and create in this way the 3D hydrogel. And this can be easily also combined with cells. So what we did was to combine this liquid hydrogel with cells, with different kind of cells, stem cells, or even like fibroblasts. And then we inject the mixed solution inside the body, and then we perform the bioprinting. And the shape that we gave to the structure helped the cells to remain there, to not go around, to not be dispersed, and also to, in case of the stem cells, to differentiate in a proper, in our case, muscle, skeletal muscle fibers.
So, big picture,
yeah.
You can, without performing surgery, the procedure is a little complicated and very innovative, but effectively you're putting new muscle into the mouse without performing surgery in exactly the shape that you want it to be.
Exactly. The only thing that we have done was to expose somehow the muscle, because the instrument that we used was not able to let the laser pass through the skin and then the connective tissue
Oh, I
into
see.
the
Okay.
muscle.
Okay.
But this is like a very simple technological limit that can be overcome by using other approaches. I mean, this was just linked to the fact that we were using a multiphoton microscope that is just designed for imaging, which is also the most important, I think, thing, because at the end, everyone in the lab, but a lot of labs in the world have like a multiphoton microscope. So this can be easily applied.
In the paper, you discussed not only muscle, but also bone and brain.
It was brain and skin. We
Oh,
didn't
right.
work
Sorry.
on that. Yeah,
Skin and brain. And
yeah.
so did you inject stem cells that differentiated into brain cells?
No, we didn't yet. It's something
Okay.
that we intend to do.
I see.
But what we observed, what we did in the skin, for example, was to deliver some fibroblasts. And what we observed was that when we created some structure, the fibroblasts were aligned in the shape that we gave. So basically, we define it as spatially controlled cell delivery. So you can deliver cell in a very precise way within the space and the area that you are interested in.
What happens to the excess if you inject too much and you don't need all of that for your 3D structure? What happens to the excess?
It doesn't really matter, because this compound is very biocompatible. So in a few hours, it's just diluted into the interspace among cells, and it's gone. It doesn't create any problem. And this is actually what happens, because when we inject the hydrogel, this is in a liquid form, and it creates like a drop within the tissue. And then we work within this drop to create the structure. Then the rest is just eliminated by the organism itself.
It's magic. Huge congratulations to you and your team. What's the next step? What will you do next? What are you working on now?
We were trying to apply this technique in vivo, as we were discussing before, to different projects actually, and also in vitro for creating more sophisticated 3D models, because also in the paper we show that you can create structure, hydrogel, inside pre-existing solid material like matrigel, which is like a widely used hydrogel for 3D cell culture. And basically in this way, we can help the cells to acquire the shape, or to give like then functional information to them according to the requirement that we need. So these are the most important perspective that we have at the moment. But then of course, everything, I mean, it's open. Let's see.
A million ideas.
Yeah.
Dr. Urciuolo, how's motherhood? How do you enjoy being a mother?
I love my daughter. She's a great person. I mean, I couldn't expect to have something more than this. It's quite complicated to combine everything, I would say. This is really the reality, but I really love to be a mom. Yeah. And also in this period, it's not easy at all. Kids are at home from school and you have to follow them. She's 7 years old.
I see.
So yeah, it's quite complicated. If you don't have the family around, you have to manage the work and the kids, and I mean, but I think that love can help in everything. So I love my work. That's why I'm like keeping on going, and I love my daughter. That's why I'm keeping to be, I hope, a good mom.
With those incredible words of inspiration, it was my pleasure to meet you.
It was my pleasure too.